Hi, everybody. I'd love to introduce Jinyun Tang from the Lawrence Berkeley lab. And he's working on extending, essentially climate modeling into ecosystems and finding good parameterizations of those models to capture the dynamics of ecosystem and in their biochemistry. Welcome, Jinyun.
Okay, yeah, so great. Thanks for introduction and for having me have this opportunity to talk about my research. So as Greg mentioned, my research is about I'm broadly defined as a climate modeler, but my focus is really on ecosystem biogeochemistry, and with a goal to trying to robustly simulate how ecosystems will fare out in the changing climate. So as you see, the ecosystem biogeochemistry is really important component of the whole ecosystem. As you might heard of, like these days we use Earth system models to predict the climate. So and the ecosystem biogeochemistry it helps to close out the energy and the chemical cycles of the whole Earth system, particularly the carbon cycle, which we think is driven the current warming of the global climate. So, but anyway, even though we have spent decades of time, we still found a lot of uncertainties with the ecosystem biogeochemistry simulations. And here's two examples from the um, climate models that the assistant models that are joining the last IPCC, the sixth IPCC assessment. So if on the left, you see that the historical simulations, you see like starting from a 1850 and towards the contemporary period, you will see that the simulated carbon sink from the land is diverged very significantly, even though they capture more or less the similar contemporary trend. On the right is about interesting topic about carbon removal. So which we think like say if we remove enough carbon and we hope the climate would return back to the more um, more stable climate in the past. But unfortunately, it turned out that all the climate models, they don't quite agree. And so from this regard, we say there's a huge uncertainty in the ecosystem biogeochemistry in those Earth system models. And people have, and then because of that, then we run into difficulties about like, say, how would we attribute the events to some, through some kind of causal link towards some kind of reasons. And the other one is like, how can we take effective action to mitigate climate based on predictions? All the things become quite risky or uncertain because of the uncertainty associated with the predicted ecosystem response to climate change. And okay, so for this, people trying to understand, spend a lot of time trying to understand why, and they have identified a lot of reasons for that. For example, for our land modeling people, ecosystem modeling people, we like to blame the atmosphere people. Their models are bad because, and that's a legitimate criticism because they still have a hard time to accurately simulate the cloud process and aerosol process. And the second one is, as usual, you'll see for those dynamic system modeling. So we always have uncertain initial and boundary conditions, and which is something it's kind of hard to get out, but unless maybe we can alleviate that through the continued collection of measurements. And the third one is for land model itself. So the land model parameters are not sufficiently observation constrained, and this one is somehow related to boundary conditions. And but the main reason is because observations are really expensive to collect mostly, and the globe is really covering the global covers a lot of different heterogeneities, so usually it's impractical to get all those observations constrained the parameters. And then this one more is land model implementation is flawed. This is a less this is one issue people paid less attention to that, but we found like because of the numerical schemes, so it's possible to turn a good mathematical formulation actually into something not so good model capability. And the one I'm talking about is the model structure. So this one is people are very easy to raise that question, but then the people really have no consensus why the model structure is uncertain. And here's why I give one examples. For example, here I pull up about 10-ish parameterizations for the soil moisture and So temperature, sensitivity, and moisture dependence for the biochemical reaction rates, such as like say how fast the soil are releasing carbon dioxide by microbial dynamics. So as you see here, we'll easily come up like about 100-ish combinations for different models to use. And with that, and you will see things that the behavior of those different parameterizations are quite different. So it would be natural to expect when those combinations are put into those system models, and we will end up very uncertain ecosystem biogeochemical simulations. So, and then there's similarly for other processes as well. For example, for stemmatic conductance, which associated with the water, exchange between land and atmosphere, and also the photosynthesis. And then there's more detailed about carbon-related processes such as methane production and the plant productivity-related nutrient uptake and microbial growth and all those things. They are similarly or equally as uncertain as those things, because people just randomly come up a lot of parameterizations without with some very specific data to parameterize that, and then once those things throw into the models, we end up something scenario like this. So if we just randomly pairing, different organs such as a head, eyes, legs, whatever color things, we can end up a lot of different kind of animals. And currently the ecosystem process that represent Earth system models is pretty much like this. Some people actually using. Oh, excuse me.
Yeah, can you actually shed light into how these come about? Because they're like stomata. It very much depends on the environment and the species and that kind of stuff. So you can't really account for all the possible species that may exist everywhere. So what do people do in practice?
Well, people just derive those empirical relationships, and then they do that by dividing the whole plant biota into different plant functional groups, such as like say in the high level, they say that's grass, but of course they can divide like say drought tolerant grass versus like say wetland grass. And similarly for trees, there are conifer trees, deciduous trees, and all actually evergreen trees. This kind of high level parameterization. But the things like when they try to parameterize that, they just take some very small sample of data set to constrain them and to derive those formulations. And those formulations are by and large is regression based. It's not like physics, they have a good reason for doing that, but they don't have good reason of that.
Okay, yeah, that
good?
makes sense. Yeah, thanks.
Yeah. Okay. Yeah. So okay. So I won't spend too much on it. What I say is that there's a lot of unreasonableness because of the possible combinations in ecosystem biogeochemistry models, and then then we studied like say we found like the fundamental reason for that lead to these ambiguity is because there are two types of models very popularly used in building those ecosystem models. The first one is the multiplicative model, which we will see also in other fields, particularly when people trying to use regression to fit or to predict some process. The second one was the law of the minimum. Perhaps you see more in some resource-driven kind of dynamics scenario. So, and because of those things, for one, because those two approaches are very easy and give everybody and every data a chance to be adopted into those models. But then the bad things they don't actually involve sufficient causality, and also because which we think is not quite realistic, because we think our world is, in many ways can be understood with physics, chemistry, all things, and a strong feature of those things is they have a strong causality they have a strong causality or information flow inside that. But those structures are not well captured by these two models, which are used to build those biogeochemistry models. And then that's end up like say, yes, we can build a model very fast, but meanwhile, it's not quite healthy, just like those junk food. But meanwhile, we can cook very carefully and considering the balanced nutrient, but then take way more time to create a better cuisine for that. So, okay, so then how do we address this question? And the way we take is like we need to look back, how to sort of dissect the problem itself to understand the scales and the process inside that. So here's a scale hierarchy we can find, or we can carve out for ecosystem biogeochemistry. And what we see is like for biogeochemistry, which is really driven by enzymes and the molecules, which operates at a very tiny scale, and for biophysics, and then this, above that, there's the biophysics, larger scale wind patterns and temperature patterns and all those kind of work patterns, and then like the ecosystem dynamics by and large, and which directly interact with management, and then finally climate. But the things like we see, what we can measure is really at this scale, and at least fine scale for ecosystem biogeochemistry is really hard to measure. But then what the model really need to resolve is this scale, and for that reason, it leads to a good explanation why the empirical based approach, which use the multiplicative model and the law of the minimum model, and that's being adopted by the majority of the models that we use to predict the climate change and ecosystem climate interactions. They focus on this scale, and they totally lost this more fundamental scale where the basic processes they are interacting. So our argument is that by going into this scale, we are able to actually create a better parameterization based on our understanding from physical chemistry and physics itself or mathematics. So and then with that, we will have a better understanding of that why those models are going to fail. Models use these approaches. The simple reason is because we know for physical processes there's scale hierarchy and also there's upscaling and cross-graining approach, the property. So what we see is those approaches, when they lift the processes from the small scale into the larger scale, they miss those. They just lose a lot of physical constraints, which are fundamental to maintain the coherence among the different process or different type of entities they're interacting with. What by entity I mean is, for example, the plant root interact with the soil and with the root, with the microbes. So those physical interactions are physical constraints, but those models, they are not able to account for those physical constraints. So which means it's naturally those models can be fundamentally uncertain and really, almost impossible to reduce their uncertainty. So, and even though we keep dumping resources and time on that. So, okay, so, and then how can we resolve this by adding back those constraints? So, our idea is to apply first principles and derive the physical rules. And then the second one is just use the mechanistic law and the construction sequence. So somehow like mathematical logics, how to combine those things and follow the information flow and or mass and energy flow. And with that, we should be able to come from the smaller fine scale and upscale to the current so-called empirical scale. So, and the approach being successfully applied in other disciplines, for example, in computational fluid dynamics, and also for industry, a lot of problems like hydrology, something like that. So one common thing is that they're based on conservation laws, and they add some phenological rules, such as, and then based on that, we can derive all those different equations coherently, but they are fitting in the same framework, which basically say we can start from the quantum mechanics and apply those first principles and get all of the things with very clear logic and understanding of approximations. So, and with that, we can even, fortunately, the whole process can even be automated or somehow algorithmized. So, and the idea is just like identify all the entities and then find their interactions, and then to identify if there's sub process, and then repeat the whole thing to eventually reduce all the interactions through our currently understanding of the transport chemical reaction, or like some mechanics. And then we start this through the recursive process. We should be built the hierarchical or more robust biogeochemical formulations. And then I was trying to show this with 3 examples how we did in the past. And I did conducted more studies than that, lot more about this, but here are only 3 examples. And okay, so let's start first example. This first example is about the trace or chemical exchange between the land and between the soil and atmosphere. So as you know, like as you see from for these gases exchange between atmosphere and the land, they are transport processes associated with from the soil into the atmosphere. And because of that, because we are driving this, it's because we are modeling this process as a gradient-driven flow. So there's the soil resistance, atmosphere resistance. For the atmosphere part, is pretty much controlled by the molecular diffusion theory and the atmosphere atmosphere turbulence. And in the soil, it's less well understood, even though they have fragmented knowledge about that. And what people did, for example, for water alone, and people just go out and they measure the evaporation and they measure the soil water content, and then they derive, use the approach I mentioned, they multiplicatively, and then they do derive those empirical relationships. And the things we found is like everybody can go out and do their measurements, like say at a certain time period, or maybe at different places, and then we end up like here are only like 4 of them, but if you go through the literature, you can very easily find more than 100 different formulas of that. So the amazing things that for many of those equations, they actually don't agree with each other, even for the same soil. So and the way we address this question is like, okay, so let's look at how the exchange actually being done by the chemical gradient, or in physical chemistry terminology, we can actually calculate the Gibbs free energy gradient. But here, anyway, we're focusing on the water, so that's based on the vapor gradient. What we see is for the water soil exchange, there are 2 pathways, and for what liquid phase, there's a hydraulic wicking based on the soil metric pressure, and then there's also the water vapor gradient driven exchange between atmosphere and the soil. And then the interesting things like the liquid water in the soil and the gaseous water vapor in the soil, they have some kind of fast exchange to establish the equilibrium, which manifests the water vapor pressure. So with that, we are able to use the Fourier's law of diffusion to create this equation, which balances atmospheric fluxes with the gaseous fluxes and aqueous flux. And then by applying our knowledge about from the Darcy's law and diffusion theory, and also some kind of parameterization of the soil matrix potential, we're able to come up with more mechanistic equations. And there we can find like, okay, so how pH going to affect the overall process. So for example, the balance between the water and the liquid water, the water vapor, and then ionic strength, and then also the soil property, for example, soil structure going to affect those parameters, and also those parameters as well. So then we'll see the actual soil resistance is really a emergent property which comes from a combination of many different factors, and not only with the soil and the soil water content. So, and then we're able to understand how, as the soil, when it dries out, how the evaporation is contributed by gaseous flow and the liquid flow. And what we see here is like, as water, start from very dry and come to saturate. First, the water vapor flow going to be dominant that, and eventually the liquid flow going to dominate that. And then Okay, so here the things that is so-called evaporation ratio, which measures is based on what vapor pressure without considering the soil resistance, we can calculate some kind of so-called evaporation potential. But anyway, we can derive those things. The actual versus potential evaporation, we can calculate this ratio. And these are very important parameters people use to measure and use to back derive this soil resistance. And then we can mimic how people do that in the field. So, but then we're also considering, because we have uncertainties in the aerodynamic resistance, because this is something cannot be reduced to zero. Some uncertainty cannot be reduced to zero. And we follow that approach. And here's one measurement people did in 1994. And they say, okay, they derive this rate and they found the soil resistance is something like this kind of curve. And interestingly, for our analytical approach, we can add a 5% random error, and we're also able to get this curve. But we know this curve is a error contaminated because the uncertainty is the aerodynamic resistance, but which reality is the actual curve should be this. And the other one is that we can play with the amount of data used to derive soil resistance on things. And then what we found is that even for the same soil, and depending how people do the experiment, they can derive all kinds of different soil resistance curve. So which indicate empirical approach is fundamentally insufficient to address this question. So, okay, so with that, we know, like, okay, so because of,
Yeah, so that's this approach obviously deals with things like conservation of energy and mass, and that's actually naturally baked in. But now you have all these parameters you have to estimate for all the different kinds of soil types and environments. How can how do you plan to estimate them across the world?
Okay, so that's been done actually in hydrology. So for example, people have so-called pedal transfer functions. So they're doing those kind of soil, how to say that? There's some people call that imbibition problem, or like drainage experiment. You take a soil and you put into and put the water into that, and let it drains and reaches like some kind of equilibrium, and then you can keep draining those soil and measure the soil water retention curve, soil water matrix potential versus moisture. But meanwhile, you can also characterize those soil with their texture. For example, if one fraction is a sand, one fraction is a silt or a clay, and then they are also characterized the organic matter. And based on those physical properties, people have built a quite robust so-called pedal transfer functions to estimate those key parameters. What I mean is the key parameters are basically the relationship between this parameter, this one, so suction pressure and the moisture, and then the hydraulic pressure as a function of moisture content and soil texture. The uncertainty is much smaller because people even derive their first principles expression mathematical theory, and they compare those pedal transfer functions. They are qualitatively very similar. Even though we know, also for that, we know actually which part of the world could be uncertain and how to reduce those uncertainties. But then for these soil resistances, because it's combines so many different factors, and taking the empirical approach, it just make it the uncertainty. It's very hard to manage.
Got it. Yeah, it makes sense. Thank you.
Okay, so here's what we learned is like, what do we see? Regression-based those parameters come up really emergent parameters. So, but they are not. Those regression cannot take into account so many factors in a reasonable way. But the first principles allow us to understand and how those parameters emerge and then actually emerge, and they also give us the opportunity how to manage those parameters by going those to explicitly to those controlling factors. Okay, so and the second parameter one example is about the affinity parameter. And if you are doing anything related to enzyme, you might recognize these equations, so-called Berkeley equations, which play a significant role in pharmaceuticals. For example, like the drug delivery modeling, and also in environmental chemistry in general. So it's about like how a chemical reaction driven by substrate is controlled by the substrate availability. So the whole thing is like for this parameter, and people use this equation to model a lot of process from photosynthesis towards microbial decomposition of, for example, nitrogen or like say organic matter. So and then this parameter is because it's appears so in many environments and people people are. Well, it's believed to be different depending on context, but then because so many models and come up and so many people are applying to that, and then these parameters become very essential. But unfortunately, parameters itself is very uncertain because it's really have a strong context dependence. Here's what we found through some kind of literature review. So usually it's a predetermined parameters through calibration or from the literature. But if we look at literature, the variability for a single process could be varies like very large, like 3 orders of magnitude is kind of common variability. And then there's a lot of uncertainty associated with like, how this parameter depends on temperature, moisture, and pH, all other things. And then we're trying to address this question with first principles. So, and what we found is, okay, so we go back to how these equations actually derived. What we found is like in the chemical reaction theory, we found, okay, so there's a diffusion-based model, so-called Smolensky model and reaction model, which fits really well and can combine with, there's quantum mechanics about reaction theory and things very neat, very consistently. So we apply this approach to the soil. What we recognize in the soil there are hierarchical structures from the, a soil bulk cube, and then within that there's a lot of like small micro environment where a lot, it's just like, for example, for humans, just like a house, there's multiple people living in the same house. And microbes do the same thing. So we can create those small micro colonies, and which actually can be characterized by their relationship to water content, which can link to the Peter transfer functions associated with the soil texture, all things, water availability. So and we start macroing the multiple microbes, and then within that there's a single microbes, and which carries on multiple transporters to take up substrate. And the whole thing is like we can apply diffusion theory towards that, which then upscale that towards from single microbes towards the whole soil structure. And within this process, actually, we can conserve the energy and conserve the mass pretty well. So, and with that, we were able to come up this equation eventually. So, we found that K parameter is actually quite involved. So it's a combination of many properties. For example, I can make a few of them. For example, for microbes, when applied to microbes, it depends on microbial cell size, transporter size, biomass density, a specific, like, say, uptake rate for a certain transporter and the transporters among how many transporters a microbes could have. And then also substrate diffusivity, where it depends on the type. For molecular, for the different size of different substrate, they could have different diffusivity. And then finally, there's a property of the soil itself. So texture, soil organic matter content, aggregate formation, all of things. So from those parameters, we see, it's a lot of the K actually emerges from so many different complex factors. And then we applied it to predict some species, chemicals. Here is an example for gases, methane, which is very important to simulate the methane global methane dynamics. So in the literature, you found like say, okay, there are at least two orders of magnitude parameters. And what we see is like from very dry towards very fully saturated soil. Indeed, we see a strong variability, and more interesting, we see as a moisture. The soil increase from very dry towards very, very wet, and the affinity parameter actually increases, which is consistent with our expectation. And similarly, we found like, okay, so the abundance of microbes and abundance of soil organic matter will also influence the parameters. But the more interesting is about solutes, aqueous solutes, which means, for example, you have nitrate in soil, or like say dissolved organic matter in the soil. And if we look at their parameter, they are K parameter, it varies actually, for example, here we show at least 5 orders of magnitude. And even for aqueous aquatic environment, people, Button, he did a survey, he found like at least 2 orders of magnitude. So depending on the factors as, type of microbes and cell size and, pH or other things. But one thing we see, like, yes, it's not because of different combinations. It's not surprising to expect people doing empirical measurement that would infer high order of variability for this parameter. So, and then, okay, so how our approach gonna lead to when applied it to somehow realistic models. And here's one example. We trying to simulate the moisture dependence of aerobic microbial decomposition for organic matter. And basically I focus here is oxygen exchange between the soil, and for this part is so-called respiration demand of oxygen, and which can link to some simple biogeochemical model. But anyway, so one thing's like, yeah, we can take the aerodynamic resistance from turbulence theory and from the resistance model from the first example, and build up and then the K parameter from this example, and then we build up a model of that. And then we can predict how soil respiration respond to the change of soil water content or dependent soil moisture content. One thing is like, we just take those parameters from literature, which measure those different parameters, such as diffusivity and soil texture, and the type of substrate, amount of substrate. We just take that. Without doing any calibration, we are able to predict very, very decent agreement with about 30 different soils. So, which is something really hard to achieve with the regression-based model. And, actually when the regression-based model, they would have to fit one parameter for each those soils, which would end up a lot of uncertainty. But we just take what's available from measurements. We can achieve very decent model data agreement, which shows the strength of this first principle based approach. So from this, we know is, okay, so the K parameter used in soil biogeochemistry or like environmental biogeochemistry in general should never be expected to be a single value. But it's really highly variable. And one thing we found, if we consider the free thought process, it could be 15 orders of magnitude variability, which is unlikely to be captured by the limited number of measurements. But our approach is able to explicitly consider, all most relevant characteristics of different entities such as soil, microbes, and even the approach can expand towards roots, and then which allow us to build a coherent framework to further introduce to analyze how temperature and moisture affected on plant soil microbe interaction in the soil, and eventually how those climate factors such as temperature and moisture would affect the global emission of CO2 or greenhouse gas in general. So okay, so and then do you have any questions?
No, it's good, fantastic, thank you.
Okay, so this is the last example. So this is a multi-nutrient regulated biological growth, which is very typical things and very important process to be modeled for any kind of biogeochemical model, either for medical use or actually for aquatic systems or also for land ecosystems. And perhaps you heard of that is like, say, there's so-called the Lebesgue's law of the minimum, which it's derived, can conceptualize it from the bucket holding the water. So we basically say the water holding capacity is really determined by lowest word. But then the question is like, does the biogeochemistry really operate on this principle? And perhaps not. So and that's for a good reason. So okay, so and even for biogeochemistry itself, for example, people in ecology, they actually did not find enough support for this law of the minimum, even though it's used for the majority, I would say more than 90% of the research published on this topic about how nutrients regulate biological growth. Okay, so and the way we attack this problem again is go back to the basic chemical reaction theory. So, and then we consider simplest example case where two substrates and one enzyme is processing or organism we can consider simple or enzyme operates on these two substrates, and we consider how they're bound together and then form this final things, the combination, the so-called enzyme substrate complex, or actually organism substrate complex that leads to the production of new biomass or product, bioproduct in general. So we can apply the law of mass action to this problem, and we can very rigorously formulate equations to derive how this final production rate depends on the concentrations of substrate A and B. So, and then one thing we found, because this allows us a very comprehensive exploration. So we know from the mathematical theory, like say, we can do various approximations. And the one thing we found, we can derive at least 3 different models, sort of like ballpark, good approximate, with different accuracy approximating original problem, what's called law of mass action. The first one is the synthesized unit model, and the second one is additive model, and the third one is the law of the minimum model. So from this graph, we see, okay, so for the most accurate comprehensive approximation, we see the major pathways is maintained. But if it comes to law of the minimum, it's get cut off from here. So the last one is really matter. So which means like the law of the minimum really missed some important intermediate information flow during the construction of this biochemical process. And then here's some
about this one.
Okay, excuse me.
You're not getting into details of organism, metabolism, metabolic structure. So to what extent are these models, like, or the combination of these 3 models, how much do you have to know about the organisms themselves?
Okay, so that's an interesting question. I think that also depends on the context. So that's one question. I mean, one topic I've been explored quite a bit. So one key idea is like, how can we upscale coherently from a single chemical reaction and towards a whole organism biological growth? So, and the idea is like we take, one thing we found, like through the law of mass action, we found it's a scale invariant. So which means in some way, if I were fixing the internal connections through the substrates and enzymes, the genes, and then so within that intracellular cell, we are able to come up more or less a coherent, for example, within single substrate growth, we are more or less come up a growth like this, just like a mechanism, which justifies the sort of like supports the idea why in the early studies of microbes, people found the so-called, at least kind of, for example, the monokinetics, perhaps you heard of that, or enzyme, or like say, mechanism kinetics, that's applied to a whole population of microbial growth. I mean, specifically there's a bacterial growth. So mathematically, even though we haven't shown, how to say that, because for one is we don't know how many reactions are there, and the second one is that the system is too complicated. But then what we found, they are mathematical invariants can be identified that sort of like intuitively support that the whole thing, this equation can be applied to a whole organism.
Well, but now, how do you know which organism? Because you have inputs A and B. Every organism is going to have,
Oh, okay. So A and B are substrate here. So, for example, like say, I will give examples. So, application examples. But like for example, for algae, what you can say, okay, that could be some kind of vitamin 10, for example. The other one is phosphorus. Or actually, for plants, it's taken up like inorganic nitrogen, and then A, which be considered as A, but the B could be like inorganic phosphate. But
it's not like your approach is to use pairwise relationships like this, which you hope to measure across families of organisms to know which let's say, model type is appropriate for each pair. Is that fair?
Yes, in some way it's fair. It's fair. Yeah, and but in any way, even for the law of the minimum, people do the same thing. So, for example, when people they're growing crops, they would like say whether my crops need more phosphorus or like need more nitrogen or actually need a new potassium. They're still doing that, but then the things like how we interpreted that matters, and the things, one interesting thing is for all those three models, they actually have the same parameters, which means the structure they're going to lead to different understand. The different structure going to lead to different understanding of the process.
Got it. Thank you.
Yeah. Okay. So and here and I just like qualitatively plot out the differences. What we see is that law of the minimum would have a cutoff of this sensitivity to the other substrate because it's just the most limiting. So and for these two models, they are very qualitatively very similar. And then so then I applied these three models and to two type of data. One is the algal growth, and which under like phosphorus, vitamin B12, which very classical data published by Droop, who based on that proposed the so-called Droop model. And then also the other one is a factorial experiment, nitrogen phosphorus fertilization experiment for plant growth. And here's on the left is for algal. So we see the three models actually, they are more or less on the similar performance. But then the interesting thing for the plant, we see the law of the minimum fails badly for some of the data, in particular when we look at least the R square of the fitting. But this is the, but look at the overall fitting, maybe it's a less issue. But then the most interesting things that we look at are those parameters. So what we found is like the law of the minimum predicts very, very different parameters, model parameters compared to the other two models, which we know is they are qualitatively more similar, also mathematically more similar. And then with these like, so for this problem, perhaps we see is law of the minimum maybe can still doing well, but then it will come at the cost of incorrect parameters. Even though we know the parameters are self-depending on the model we're using, but then given our good understanding of this chemical reaction theory, and we can design some systems actually to derive those more or less true value of those parameters, and then through that we can very clearly see how law of the minimum sort of like distort those parameters and to make the model work. So that means like it will lead to definitely some kind of how to say that good results with wrong reasons, which something we really don't want to happen in a predictive model. So from this we learn is law of the minimum is a poor approximation to law of the mass action, and it has distorted the true parameters. For example, one analogy is like one of my friend told me is he's doing compute CFD. They once created a model to simulate the movement of water, but somehow they found like so they have to use the viscosity of the honey to get reasonable simulation of the surface flow of water. So it's something law of the minimum. It seems often leads to that kind of a situation. And the other thing is like for the additive model, what we found is it's really scalable, so we can very easily add in more nutrient, and also we can give those parameters a very specific physical meaning. So and it seems from this, it's from this model where I can upscale reasonably towards the whole organism, from single chemical reaction to organism. Of course, a better argument would be based on law of mass action itself. But then from there, we also find actually there's mathematical invariance. We do something similar like this. Then this justifies like, why the early studies of bacterial growth, they can derive the monodic growth curve, which in many ways like this final form. So to summarize for the power of first principles, so it's based on our current knowledge of physical chemistry and physics and perhaps mathematics as well, I think we're quite ready to re-tackle the biogeochemistry problem and turn a lot of formulated process representation into first principle based or like physical rule based. And because if we do that, the result model can integrate many more environmental factors more explicitly rather than just based on regressions and which, unknowingly sort of like prune out a lot of relevant influences from those combinations. And because if we're doing that, the prediction will become more variable. And the one criticism of current ecosystem models is they lack of extrapolation capability, particularly when we're varying the climate, varying the soil parameters. We don't see much variability actually in the predicted, ecosystem fluxes, carbon stocks, and something like that. And then the final is like with first principles, because we can have a more mechanistic understanding, and it will also provide us better opportunity to manage those ecosystems we are trying to simulate. Okay, so then how to apply that? Our idea is like we need to recognize their hierarchical structure exists on the ecosystem process, and from which start from really small scale where biochemical reactions occur towards very high level. Things are subject to human management, like fire or all kinds of pathogen past attack, all of the things. So if we apply the physical rules at those scales, and then we use some coherent rules to upscale, then we should be able to apply these develop those models iteratively. And then the key things here is really, so as we are doing those upscaling, just to make sure we are carrying those subscale, fine scale physical constraints sufficiently from when we are lifting this small scale towards a high scale. So we start, and okay, so that's all my talk today. So thanks.
Okay, thank you very much. Well, one, I guess, application of your thinking here is there's a lot of let's say crop simulations because people spend time and money on crop simulations, so they mimic your more first principles approach is closer. I mean, they have their own empirical relationships as well, but to what extent can you start leveraging these lower level models within these higher level ecological simulations?
Okay, so I would say hydrological simulations, I think it's pure hydrology perspective. I think they are in their studies, I would say people have less ambiguity about how those equations should be, even though at a really fine scale, then eventually, because they have a mixed flow rate, which some of them could be described by better perhaps by the, how to say that, Navier-Stokes equations, or actually some simplification of Navier-Stokes equations, and then some larger scale of the recharge flow, or a combination of the two. And for those things, I think one approach is like based on that, people come up with fine scale approaches and then identify those factors, and then to do cost grading by mimicking what people done in the turbulent closure. I think that's being attempted by a few hydrologists. But what I'm saying for ecosystem biogeochemistry, I would say, particularly for crop modeling, their situation is pretty much like ecosystem models. So that if you dive in those models, you found a lot of representations actually very empirical.
Yes.
So yeah, so my recommendation here is actually to rebuild those crop models, particularly for the biogeochemistry part, from the nitrogen cycle, carbon cycle, and towards the overall growth of the crop and the tillage and those kind of a drainage management through this physical rule-based approaches that will give us a more confidence on ecosystem on crop management than in terms to maintain that yield and meanwhile the soil quality.
So now the motivation for doing these coarser models, I mean, you're right. So these like D sets and whatnot, they're very they're fine grained but empirical.
Yeah.
The reason why it's availability of data, it's availability of human labor to cover every species. I mean, it takes many years to even get like a corn or a strawberry, and we're talking about some pine trees and scrub brushes, many more species that are relevant to the climate. So, to what extent can we automate this process, maybe make it more data driven?
I would say, for one, if you're really data driven, it's like we are facing the reality is it's really hard to connect sufficient data. So that means like we need some good, high confidence, or like say, how to say it, high resolution model, or like say high confidence, like detailed model to provide those data, to pre-train, to generate those data, to build, and then which supports the build of data-driven model. But in the short term, I would say we still lack of this kind of more or less first principle driven model. So the one I know is one thing I've been working on that kind of close to this is the ecosystem model, or like the one model I'm developing is so-called ecosystem model, which is very systematically apply those approaches. But even with that, because of I think because of the how to say that the amount of effort need to be put into that, and the historical people only very few people have put time on this, and we also haven't seen a wide applications. But for ecosystem model right now, there are more people start using that. But again, we haven't built those kind of a data reservoirs, whatever you call that, like data portals, to generate sufficient data actually to drive a data driven to derive a data driven model for larger scale.
Right. And now, probably the closest analogy I can think of to what you're doing is like whole cell models. It's in E. coli, you have a giant, incomplete metabolic network. So I guess there's this. You can go crazy with the complexity, which if you go whole cell model, that's simple enough. They actually really do try to capture all the chemicals that are sloshing around. I guess what's the resolution that you would have in mind? Like for, let's say, a pine tree should be a mainstay of carbon absorption, or like Amazonian palm trees, that kind of thing. How many dimensions of data collection might be required to make a decent enough model?
Okay, so I think what my argument would be like the advantage of the approach I'm trying to do is because I think it is coherence, the structural coherency. Because in mathematics, for example, in calculus, perhaps you've done that, like say numerical approximation of some integrals, and based on the discretization, so of the graded size, we can quantify the uncertainties, and we're not necessary to resolve every corner of that, even though there's could be singularity point, but like say in the calculus, it's less important. But the same idea when applied to the model a biological cell, I would say is if the mathematical structure we build up is a self-coherent, and I would say that the resultant model will be more resilient to those uncertainties associated with the missing of certain chemical reactions. But then meanwhile, it does provide a estimation about what's uncertainty are there, because we can, based on known chemical reactions, and take away some of them, adding some of them, and quantify the uncertainty of that. And that's because within the same coherent mathematical framework. That's going to be much better than randomly plugging these regression empirical curves. And then, okay, so then come back to the plants for the tree, for the pine tree. I would say in the basic level, we should be able to represent the cannabis structure to some extent. And also, and then to the growth cycle, we should be able to capture from the seed towards the whole mature adult plant. And in the soil, we should have basic morphology representations of roots, basic key structures, like, for example, primary roots, taproots, and fine roots. And then because those things are actually measurable, we know their physical morphological characterization, like say how thick are the bind roots or like cross roots, their relative biomass things and how deep they go, and the elasticity, all those things. If we have a model represent the detail to this kind of level, then that way we have a better chance to directly link our field measurement campaigns. And then that will be able to provide, for example, like that enables more data streams from the experiment side to be assimilated in those models. And a second one is because the model is representing a tree, it's really like a tree. It's not like, many counter models just doing like the canopy is a triangle or actually a cylinder, and the plant is a tube. So which of course is unrealistic. And then we should not expecting actually fit the measurement of a real tree towards some kind of tube. Right. And that's the problem with current approach. So what I'm saying is our approach, my approach is like we should take advantage of the coherence, structural coherency, which we've seen in mathematics. Like, you study, particularly for calculus, that's a really good example. Or like numerical analysis, numerical simulation of those PDEs.
Right. So then, actually, what you're describing sounds a lot more like DSET, AppSim, process-based models. And they do have components. I mean, I'm thinking for annual plants, more crops, they have less data intensive, where they have roots and leaves and stems and flowers, and those are separated out into separate modules. But now, and so you can imagine building upon that for all plants. But is real world data. So if you're doing test plots, which agriculture they can, you can do it. A lot of times you have satellite observations, occasional pass through with a drone to get tree heights or chemical composition. That's it. So how can you work in that data sparse environment?
Well, for data sparse, I mean, the other things I should mention is like the approach I'm taking is, for example, I use heavily so-called physical laws. And then that allows us to leverage a lot of measurement from other fields. For example, for those kind of sewage treatment bioreactors, because they're more or less, for example, particularly for microbes, because the microbes, the biology, more or less, they are same in different environment. Mm-hmm. And then because we're facing the data sparsity problem, but the processes are more or less operating at the same, at the different places. Mm-hmm. So again, if they take opportunity, take advantage of this existence of a huge stream of data in other fields with this approach, then we are at a better chance to extrapolate to regions even without much data.
Yeah, no, it makes a lot of sense. Thank you. All right, cool. So we're at time, but yes, this was fascinating talk. Thank you very much for joining us.
Yeah, thanks.
Oh, have a great rest of your day.
Yeah, have a great rest of the day.